Welcome everyone to the seminar of the IF. The topic for this one is Understanding and Conserving Salmonid Populations in a Changing World, Theoretical Underpinnings and Implementation. And it will be given by Dr. Bruce Stockley and Professor Jamie Stevens. Dr. Bruce Stockley is an applied river scientist who uses evidence-based approach to conserve and restore rivers in the southwest of England. Has a background in fishery genetics, morphological, molecular, and paleontological approaches to the understanding of ecology and evolution, and is currently Director of Operations at the Westcountry Rivers Trust. Professor Jamie Stevens is in the Department of Biosciences at the University of Exeter. He is, on the other side, a molecular ecologist and evolutionary biologist, and his research focuses on the application of population genetics to explore questions in ecology, management, and conservation. So, sorry for the delay and the rush introduction. So please, Bruce.
Thank you very much, Emilio. So as Emilio's done his introduction, my name is Bruce Stockley. I thought it'd be really interesting to give a presentation by two people, myself and Jamie Stevens. We've worked for more than 10 years together to understand that both the theoretical understanding of fishery population genetics for salmon and trout, and then for my side, the implementation of that for conservation work. And I hope this provides some interesting insights as to how an applied scientist like myself can work with Jamie, who's looking at the much deeper into the population genetics of these species. So if we turn now to the area of interest, so that is the West Country, the southwest of England. If you look at this map, you'll see a wide range of different rivers of different sizes. Now, all of those rivers will have trout in them, and most of those rivers will have salmon in them. And as you can see, it was quite a large geographical area. But unfortunately, particularly for the salmon, they're not doing that well. So my mission to conserve and improve those populations is really challenged by the fact that those populations are at risk. So if we look to the assessments by CFAS and the Environment Agency, you can see with great confidence this map suddenly turns red and orange, and as most times when that happens, that means things are not good. The salmon populations are at risk with a very high confidence level. And in the few areas where you see they're not definitely at risk, they're probably at risk. And the trout populations are following on behind there. So what we have is a rather serious situation of looking at the potential extinction of these species, which undergo some of the most incredible migrations in the animal world. Now, to explain some of the causes of this and what we can do about them, I'm going to use a bit of an imaginary river, which is called the River Zed. And if we also turn the clock back 100 years, we can look at what did this river, this typical West Country river, look like in 1920. So you have this small river, you've got 5,000 salmon coming from the North Atlantic, trying to get up to the top of the catchment. And there they'll spawn, they'll create nests, and then the next generation will complete the cycle so that they can then go to sea and the species continues in this particular river. Now, this being 1920, the various weirs in there were not optimally designed, and we might say they have a 75% passability rate. So what does that mean? It means that if 100 salmon try and get over that weir, only 75 will. And in this particular scenario, we've got 3 of those weirs. So when you do this repeated, only 75% get up, and of those remaining, only another 75% get up, you then up with about 2,000 fish that get up to the top of the catchment. Now that is enough. That's enough fish to spawn, to fill all the habitat there, to lay all the eggs that could be laid in that particular part of the river, and then the cycle continues. So we can say it's unfortunate maybe that not all the fish could get over the weirs, but it worked. It worked back then. Let's roll that forwards to somewhere approximating the present day. Now we don't have 5,000 salmon coming into the catchment. We have 500. Now, some people have said to me, isn't that a bit of an exaggeration? Really, is it that bad? We've really got 10 times fewer salmon coming into the river. And so unfortunately, yes, we have 2 fish counters in the West Country, and we can confirm that these are the kind of declines in numbers we're looking at. So these are we can treat them as real numbers, as realistic figures. Now, as you can imagine, as you do those numbers, you can find that you've got 500 fish coming in. So you don't have 2,000 fish getting to the top of the catchment. You've got 200 fish to try and populate this this river for the next generation. And half of those will be males. So you've only got 100 spawning salmon in this river. And that's just simply not enough to create enough nests and lay enough eggs for to fill the river with the next generation. And not only that, when they actually do get there, the rivers have there's silt coming into the river, which smothers the eggs when they're trying to develop. There's overshading, so abandoned coppice in these trees links together and it shades the river completely. So what that means is that no light gets to the river, and almost all ecologies in the world require light as their foundation. So you have no light, that means you have no invertebrates, which means you really have very struggling juvenile fish. So what this sounds like a story of downward decline, a downward spiral that when you look at those numbers seems almost impossible to get out of, but it really isn't. The work that we've been carrying out, and many other rivers trusts across the whole of England and beyond, if we take the first weir and change it, we improve it from a 75% passability to a 98% passability, and then the middle weir here, weir B, we remove it completely. So that's 100% passable. The fish can get right over there. And we make the next weir into 98% passable. We then have nearly all of our 500 fish that got into that catchment. They can get to the top. They can get to the top of the catchment. So we have improved the fish passage. So all the fish that want to get up can get up. And then we carry out work to improve the habitat so that when all the juvenile fish that are there, they then have the best chance of becoming next generation and continuing this cycle. We only have, like anyone, only have limited resources. We can only do this in certain places, and we need to know where we need to do it. We need to know where we're wasting our effort and where this is useful work that needs to be carried out. And we also need to ensure that whilst I spend all my time thinking about the rivers, I'm not forgetting about the North Atlantic, where all those fish are going. So those are the kind of questions that I go into when I try and carry out my work to improve the rivers for salmon and trout. So I'm now going to hand over to Jamie for the central part of this presentation.
Okay, so you should have a fairly boring slide there with a lot of text and white space, yeah? I can see that, good. Thanks, Bruce. I like your use of the River Z. I once gave a talk where I talked about the River X and the River Y, and as someone in the audience pointed out that there is a River X and there is a River Y. If you're based in Exeter, the X is very much a river on your doorstep. Anyway, I digress. So the scene that Bruce has set there, and we work quite frequently with the Westcountry Rivers Trust and various other organisations focused on the conservation and management of salmonids in the West Country and beyond. Oh, my slides have started moving. Yeah, so that's an introduction to the question. One of the things we often want to do when we have to look at fish moving in rivers is not just understand how many fish. That's kind of, a lot of the original efforts to understand whether a river was doing well was about how many fish there were in the river. We've got better at that, I think, as a group of people involved in management and conservation of salmonids. And it's not just now about how many fish, but what is the diversity of fish within those systems. And that's a way that we can move to a much more subtle assessment of the quality of the fish within our rivers. So, as I say, initially, excuse me, a lot of the effort would have been about counting fish, and that is still the case. Fish counters are employed on a whole bunch of rivers, index rivers around rivers within the United Kingdom and equivalents within France and things like that. But there's a realization now that we also need to be looking at preservation of diversity, and that's perhaps where molecular methods can offer some ways forward that simply counting things can't always do on its own. So that's a little bit what I'm going to talk to you about. I'll give you a bit of background. So traditional approaches to identify discrete population units, often referred to as stocks, a stock of fish. You might have heard people use this idea, this term about a stock of fish. If you actually try and pin someone down, you'll find that whoever you're talking to within fish and fisheries, there are many different variants on what the word stock means. Typically, we use it to talk about something that has got a relevance in time and space and perhaps life history characteristics. But people will often talk about a stock of fish that moves early within the season, and there we might be talking about an ecologically driven movement of fish earlier down a river or indeed up a river, or we might be talking about something that has a genetic basis. And all of these things can be used and have been used by river managers and fisheries managers. So initially things, and in some cases still are, would have focused exclusively on phenotypic variation. They might have looked at scale characteristics or morphometrics. All sorts of things have been used, and isotopic work. More recently, and indeed ongoing, there's lots of effort on use of tags. Tagging can be extremely successful, where you've got a very confined set of fish that you need to understand their movements within a river system. But the downside of tagging is that you have to catch the fish. You have to catch them at a size when they can hold a tag. Catching and tagging a fish is stressful in its own right. And then there's also the obvious factor that we need to recapture them to be able to either count them, or in the case of tags that have some capacity to measure temperature or depth or other factors experienced by the fish, we would need to recapture that fish, and potentially that recapturing can be lethal if you're taking out a full radio tag, or at least you put the fish through another stressful event. That is if you can even recapture them. And in some large tagging studies that have then let fish go to sea, and you'll be tagging at a kind of par stage or a smolt stage, and I'll talk to you about the salmon life cycle in a sec for those of you who aren't familiar with that. But essentially, these are variants of the juvenile stage before the fish goes to sea. It's a lot of effort, a lot of stress on the fish for often low returns. So genotypic variation has been postulated, probably since the 60s, when people started doing isoenzymic studies. And this has come, through a whole range of different techniques, and to the point now where we're using things like SNP panels, or we might even be using whole genome sequencing. And that's what I'll talk to you about. And I'll try and relate that to some of the uses to answer some of the questions that Bruce has given an outline to in the first part of the talk. And I'll leave it hopefully in a position where Bruce can pick up and show how we use that. We have used that on some river systems in the Southwest. And we will talk mostly about the Southwest. That's because both of our institutions are based in the Southwest. But outside of Scotland and Ireland, and indeed Wales, a lot of the rivers that contain salmon and trout are in the south and southwest of England. Not all. The Tyne is a very big river, and there are some other rivers with trout over in the east. Anyway, I think it's important that just for those of you who don't know, the salmon life cycle and the salmonid life cycle, this would apply to trout and sea trout in the same way. The key thing about this, and the reason that we can use genes and genetics to understand diversity of fish within a river system or within a series of rivers, is because, and I'll stick to salmon, but think of it as trout and sea trout. Salmon they spawn in a river. They go through a whole range of life cycle stages in the river. They there's various ways that we think about how they might imprint on that river, whether it's the actual geochemistry of the river or whether it's their cohort population within the river that they imprint on, and there's some evidence to discuss that both things play a part. Eventually, though, they undergo this smoltification, and they smolt and they change fundamentally their physiology within the fresh water to prepare them to leave the fresh water of the river and to go out into the sea to feed. If you're a salmon, you might move hundreds, if not thousands of kilometers out into the sea to feed. And the risk is big, but the rewards are immense. The feeding within the marine environment is huge, which is why trout also exhibit a sea trout phase, and they move into the marine environment to feed. And in doing so, that the quality of the food, the shrimps, prawns, mollusks, etc., crustaceans, offer so much more nutritionally than some of the things that they're otherwise trying to feed on within a freshwater stage for things like trout that opt to stay resident. Salmon, other than a few fish which mature early, all will end up going to sea. So they complete that whole life cycle. And the critical bit, the bit that if you don't know about this, but I'm sure you will from the many nature documentaries that are produced on this, is that they come back to their natal river. They come back to their natal river with a high degree of fidelity. There is a certain level of straying, and that's almost certainly an evolutionary adaptation, because if they didn't stray, they would never have recolonized many of the rivers of the northern part of Britain after the last glacial maximum. So they need to stray a bit, but predominantly they're returning to their natal rivers, and they are breeding with other fish that have been spawned in that river. And in doing so, they strengthen the signature of the fish within that river. So it's in a human sense, it's like marrying someone from the same village. In all probability, if you've carried on doing that over generations and generations, you will get a signature of the people within that village that is distinct for that environment. And in the same way, these fish do the very same thing. So they are building this genetic signature that might be specific to a particular river. In some very large catchments, it might be particular to a particular tributary within a river. But it's this particular signature that if we can kind of read that genetic tag, we can read that genotype, and we can recognize it. If we can do that, that allows us to look at the diversity of fish within a river, and it also allows us to be able to track fish that are moving at sea and understand, in the absence of a physical tag that's been inserted through a tagging program, it allows us to understand where they might be heading back to. Of course, if we're going to use genetic signatures for that kind of understanding of the way fish are moving at sea and are returning to different rivers, we need to be able to we need to be able to assign them back to a river of origin with a certain degree of certainty. So we can look at population structure, we can look at diversity, we can look at gene flow, and all of these things feed into the kind of questions that Bruce was posing in the earlier part of the talk. So subsequently, we can also use this in a very applied manner to look at assignment of population of fish that are moving in the marine phase back to their river of origin. We can also look at fish that have returned to a river as adults and check that they are returning to their natal river. So we can look at things like degrees of straying. And lastly, we can use genetic techniques to look at sexing, which can help us to build better pictures of egg deposition limits. What do we use for these things? Well, we can use a whole range of genetic techniques, and largely, as I'm sure for those of you who do this kind of work, you'll realize that at each stage, if you look down that list and choose your favorite technique, you'll get to see that we can understand more and more about the variation at the level of the genome of the fish. So we're not just looking at proteins, which are effectively a proxy for the genes which code for them, as we might be doing with isoenzymes, but we can actually look at SNPs, single nucleotide polymorphisms, across the entire genome of the fish, or with relatively small genomes and enough money, we might go in for something like whole genome sequencing and use that in a population sense. So if I scroll down, typically though, if we want to characterize populations of fish, we need to get a bunch of fish from a particular set of rivers, obtain a genotype, and then do some population genetics on those, some statistics to look at the degrees of separation. And this is a study that we would have done in consultation with Bruce and the Westcountry Rivers Trust and the Atlantic Salmon Trust and various other people. And what you can see is a picture of genetic variation in this case, it's trout populations in rivers along the south coast of Cornwall. And what we've got is a very obvious split between the rivers in the west of Cornwall and rivers in the east of Cornwall. And we can see they're, really quite distinct genetic differences, which suggests probably a lack of strain between these regions and a lack of gene flow between these populations. But within these regions, and these regions of sort of east and west of the big China clay area, which sits around River 11 in the middle there where that red dotted line is, we can break that down even more and look at kind of localized genetic variants. And we undertake some of these hierarchical studies, which may or may not be useful. In the top one, the middle panel, the top of the separate regions, what we're seeing there is evidence that you've got quite a mosaic of diversity. And in the bottom panel, you can see that actually, apart from a couple of two rivers, we've got largely a sort of a mix of genetic signatures. And it's a reflection largely, there are exceptions, but it's a reflection largely that the rivers in the west part of this region are quite small. Some of them are very difficult access, and to relate to some of those things that Bruce talked about, some of them have very large weirs that enter at the entry from the sea back into the river. And what we see is that drives isolation, and it drives these distinct populations. So some of these hardships do drive differentiation, but of course they also result in overall low numbers of fish, which can leave them very vulnerable to pollution events and other sort of local catastrophic events. In the bottom panel, you see trout from largely what constitute bigger rivers, and what we're seeing is subtle changes across distance, and that suggests that overall, apart from the Polperro and the Par, the purple and the green rivers, we've got really movement of fish between rivers. And this might make it easier for someone like Bruce and the Westcountry Rivers Trust when they're talking about fisheries management. We can also use some of these things for assignment to population. So we do that same kind of thing, and these are some preliminary data from a much bigger study than that one I showed you. These are on sea trout moving around in the English Channel, and what we had to do here was to genotype fish from all the rivers that flow into the English Channel and a few beyond, because we might well encounter fish moving in the channel that haven't come directly from channel flow rivers. So we've gone to rivers flowing into the channel all along the south coast of Britain, all along the north coast of France, and then we've genotyped those. And what we've done is we've looked at the level of genetic separation between them, and we end up with these things called reporting regions. And this is effectively, it's a statement of larger groups of rivers which share genetic similarity. So we're not perhaps able to identify rivers down to the level of individual rivers, but we're able to identify populations of fish, in this case trout, within groups of rivers. And in terms of understanding where fish are going, that are traveling around in the marine environment, as a first kind of look at where they're moving, this is enough to help us with our management aims. And our management aims in this perspective are, where are fish coming from, where are they moving to, and where are they being exploited within the channel. So the first thing in that middle panel, you see all the sample sites, all the red dots. Then the middle kind of tree diagram there is a genetic assessment based on SNP genotypes for all the different reporting regions, which are essentially comprise all the different rivers. And if I was to blow that figure up, you would see some of the rivers that we've just talked about there in the west of southwest Britain. And then in the final panel, we've overlaid those distinct regions onto the map of the channel. And what we're able to do is we're able to undertake then, and I'll move on a bit quickly just because of time, but this is essentially a schema of that. It takes you from the in-river where you're sampling juveniles all the way through SNP analysis to the phylogeny there, and you can see the red rivers here constitute some of those rivers in the southwest of England. So in this study, we're actually labeling the southwest, so that'll be most of South Cornwall and bits of West Devon as a single reporting region. And what we can then do is the same thing again, but using a fish that has been caught at sea. And in this particular project, which was the SAMARSH project, we had some resources, some funds to hire boats to take fishermen out and to fish specifically for fish moving in the marine environment. And we do the same thing. We take the genotype of the fish moving at sea and we kind of shoot it back against our panel, and using some assignment software, we're able to say where those fish caught at sea were heading. Essentially, we're doing this. So this is assignment back to individual rivers, and this is what we hope will work. And as I've already said, sometimes it isn't possible to assign to river, but we can certainly assign to reporting region. And this is a summary of just a few of the results. And what we've got here, keep your eyes on the big blue numbers on the right. We've got 9 fish that were caught at sea around Rye Harbour, and we've got 53 fish, sea trout, that were caught in the sea off of South Cornwall. And what we've been able to do is we've been able to genotype those fish and assign them back to the reporting regions that you can see in that map of southern Britain and northern France over there in the top left of this slide. And what we've been able to ascertain is that, so for instance, for Rye Harbour, we see around 7 of those fish coming from the Hampshire Basin area, 1 from northern France and 1 from East Anglia. So it's obvious that predominantly these fish are moving in that part of the world from west to east, but there are also fish that are making quite long distance migrations, perhaps against the current and against the sort of trend that's exhibited by the majority. In the southern, the lower panel, where you see again southern Britain, the Channel, 49 of those fish were obviously sampled locally in the Cornwall region, but again we had fish coming from South Wales caught in those nets off of Cornwall. We had 2 fish coming from the north coast of France, and we had 1 that had a completely unique genetic signature. So what we're able to evidence is some long distance movement by sea trout around the southern North Sea and the English Channel. And whilst this might have been well expected for things like salmon, this was really very little known about this in trout. So that's something of a breakthrough for this project. And lastly, I'll just say that we can use the genetic methods also for things like genetic sexing. And this is a slightly different approach. This is just a very simple sequencing approach. But what we're able to show is that fish that have spent 1 year at sea have a very sort of high level of predictability in terms of their sex ratio. Whereas with fish that have spent 2 winters at sea, the actual level of their sex ratio is very different from that predicted from previous data. And what we did with this particular study was to emphasize how important it is to get precise estimates of your sexing data for the particular river that you're interested in studying. Just to finish and lead into Bruce's last phase of the project, I thought I'd show you this. This is a SNP study of some work done on the Delank tributary of the River Camel down here in southwest England on the north coast of Cornwall. And what we see is SNP genotyping of fish from all across this part of the Camel catchment. And the bit that you really want to take home from this are the green dots, which is the Delank. And you can see that they stand out on their own. They've been isolated above a quarry for several hundred years. And even in that short time, it's driven this distinct genotype. If you look in that middle panel with the two lines on, a red line and a black line, what you can see, the black dots represent fish that show their genetic differentiation relates to their geographic differentiation. And we have a relationship which I think from memory is not quite significant, but pretty much on the border of, whereas when we add in the delanked fish, that ratio, that relationship between genetic distance and geographic distance breaks down completely because they've been so isolated. And this is a nice way that we can show how we can investigate the way that fish can differ over very short different distances in isolated populations. And it makes us then leads us on to questions about how we go on to manage those fish, given their degree of genetic differentiation and separation. And with that, I will hand back to Bruce.
Right, yeah, thank you very much, Jamie. So if we now look at what is that impact, how can that make a difference to ultimately the survival of these different populations in the rivers in the West Country? So if you look at this picture here, you will see a fish processing plant in Iceland. So we visited, myself and colleagues from Jamie's team visited this Icelandic fish processing plant, and we saw how it worked. And essentially what we had is large numbers of pelagic fish, so herring and mackerel and so on being caught, and there was a small bycatch, unintentional bycatch of the intermediary stage between the large salmon and the juvenile salmon that are making their way, which happened to be exactly the same size as the fish they're trying to catch. So potentially mixed in with these huge numbers of mackerel, herring, etc., are salmon. Now, given the state of some of the rivers in the West Country, but also across the west coast of Europe, then that could have quite a significant impact. Now, this also was occurring off the coast of Ireland as well. So we have two fisheries here, and you don't need to read the references, but they are there. The point is that using the genetic techniques that Jamie has just outlined, and if you recall you have a mix of fish and then you spread them out and you can identify which river they come from. The process of that was carried out both in the Icelandic fishery and in the Irish driftnet fishery, and different results were found. So in the Icelandic fishery, all the fish found in that analysis were Icelandic. Now we know that rivers in Iceland are not suffering the kind of declines elsewhere in Europe, and therefore that is not a concern. However, when you do the same for the fish, the nets from Ireland, these high seas nets, they found numbers of fish from the West Country, which is obviously my concern, but also Spain, which has had severe, if not worse, worse declines than we've seen in the West Country. Also France and other countries as well, and therefore that is what we call the mixed stock fishery, where you are having you don't know where your fish are coming from. You can't tell when you catch them which colour they are, which river they come from, and therefore you have every chance of extinguishing a small and struggling fishery without even realising it. So you can see this genetic impact. These genetic studies are allowing us to manage the marine phase of the life cycle of the salmon. But let's zoom in a little bit more now. So let's talk about the regional impact. So as you all know, I'm particularly concerned about the southwest of England. And if we look now and we zoom in on one river called the Tamar, which is one of the largest rivers in the southwest, and that's in that dotted line there. And what happens in the Tamar is, whilst we don't anymore have a commercial netting occurring, we do have an angling, quite a busy and successful angling activities in the lower and the upper catchments, part of the Tamar catchment. Now, when the Tamar's doing fine, you may think, oh, it's fine. Okay, people could take a few fish and have them for their supper if they catch them. That sounds fine. There are enough fish in the river. Why not? Well, when the genetic analysis is carried out, and it should be pointed out that if you think back to that sort of the number of dots as you go down from your isozymes down into your full genomics, that we're moving down the scale now of the level of detail of your genetic analysis, that some of those fish, but only in the lower catchment, are from other rivers, and some of those rivers are really small, and you know you will barely see them on the map, and they may have, you know, far less than 500 fish in there. And if we were just to enable those fish to be caught and killed, you could eliminate those small rivers populations quite readily without ever knowing they were there, without some form of genetic analysis. And I really could see no other way you would do that. But the other part of the story is the top of the catchment. They are native fish that are to that, and therefore you can actually manage those as a Tamar stock, as Jamie used the word stock before. So we're sort of getting into that level of detailed analysis. So that's the sort of regional level of impact from these studies. So there you go. There is the study there. Now, let's drill down again and again. This comes down to your having to use more and more detailed genetic studies that get more that extract. You're getting closer and closer to the maximum amount of genetic information you can extract from fish, because of the genome. You can only go so far, to the local impact. So this comes to that thing I was talking about at the beginning about the prioritization. Which of these stocks are struggling? Which of these stocks need that help, need that weir removed, need the habitat improved, and so on. And now I'm going to turn to a couple of rivers, the Camel and the Foy, which are slightly smaller than the Tamar in Cornwall. So these this is an SMP, so again, a more detailed genetic analysis, and what that tells us. Yeah, and if you think back to Jamie's talk, we're talking about the Delank. If you recall, he mentioned Delank, where you're talking about those very, very distinct fish, but also when he was talking about the southern Cornwall, the sort of isolation by distance, which is part of the Foy. So what does this mean? What does this what does taking action on this local information mean? So here we have Keybridge Weir. So this is, if you think back to the River Z example, this is the middle one where we completely take the weir out. That means that there's 100% passability. And interestingly, this is directly below that isolated population that Jamie was talking about on the Delank. On the Foy, we're not able to remove this because it's an Environment Agency gauging station. It's part of our water monitoring network. So in that case, we employed these fish brushes to add. We had to extend the fish pass, put the brushes on, enabled the fish to get over. So that will take us from much up to this sort of 98% passability. The same with Camelford. We couldn't remove this one because there's a foul sewer running across the top. We have to work with how we find things. So we put a baffle fish pass in there. And now we come to the habitat. So if you recall, I said there's no point getting the fish up there if they don't have habitat which can support the juveniles. And if you look at the sort of before and after these picture, even in the winter time, there is far more light getting to the river on the right hand side, which supports its ecology. Although, as you see, we don't, you know, in the past, people may have just clear felled those to achieve that. That's really not the way forward. You need a much more nuanced approach. And of course, that produces a lot of woody debris. And by tying that into the river, we can recreate habitat that has been lost by past management, a lot more habitat diversity, cleaner gravels, and so on. On the subject of gravels, again, just below that Delank population Jamie mentioned, we because of the quarry, the way that it actually captures all the gravel, there was just a bedrock there, which means the salmon and trout can't complete their life cycle. We've added gravels into there. So when you look at that whole picture, if you remember, I was talking about the impact of just dealing with 3 weirs. The project that I'm showing you now, we dealt with 17 obstacles to fish passage over 2 rivers. So you can see we can expect quite significant improvements in the numbers of fish that get into there. And of course, those habitat improvements as well. I hope this has given you a good overview and that you can sort of keep in mind the fact that we can work, these genetic tools can allow us to manage these fish at an international level, at a regional level, and at a local level. And therefore we can get a fully integrated cooperation between that theoretical understanding and the implementation. So thank you all very much for your time and attention. And I'll just close the presentation there.
Thank you so much. So we have time for 1, maybe 2 short questions. So is there anyone that have a question? If not, I can do it myself because I have one. Okay, then I'll do one that is a little bit for both of you. So As we spoke about salmonid conservation, and we've seen different features like the morphological analysis, gene flow, genetic diversity. What would be, if we want to conserve or if we want to detect the impact of, for example, pollution or human activities, what would be the best indicators to see those first stages of alteration?
Jamie, I don't know if you want to give the example of the metal tolerance in trout. Might be useful.
Yeah, I was gonna pitch in with that. Shall I kick off with that?
Yeah.
Yeah. So Emilio, to answer your question, good question. Typically what we see, you know, if it was a point source pollution where you had a massive fish kill, then you're going to see a lot of dead fish. All right. But from a more subtle perspective, you know, one of the things that Bruce and I and the Westcountry team have been involved with for a long time is looking at legacy pollution from mine water washout, which is still a huge problem in the southwest of England. Huge amounts of mining in Cornwall, West Devon, but there's also a lot of mining in Wales and northeast of England and parts of Ireland as well. What we've seen from our studies is that we don't know quite how the fish population suffered at the immediate onset of the mining activity, and because a lot of this water was used in the mining industry, and subsequently water has flowed into mines and has to be pumped out. Of course, it's full of toxic metals that are, you know, lead, copper, arsenic, tin, all these things, although tin's not particularly soluble, but copper and things like that. That pollution, that legacy pollution is ongoing, and yet we have healthy populations in some rivers of salmon, but in many rivers we have healthy populations of trout. And in the west of Cornwall, there's a river called the Hale, where we have a perfectly functioning population of beautiful golden trout. And when we do the genetic analysis, what we see is that numerically there's lots of them, which is why I think just counting things, I think, you know, we're well aware that we're beyond that. What we see is extremely low genetic diversity by all the standard measures, you know, observed heterozygosity, allelic richness, all those things. And so what it looks like is that the fish have been able to adapt, because we've got other markers under selection that we're putting together in a paper that we're setting out shortly. Lots of some evidence of adaptation, but the first thing we see is massive decrease in genetic diversity across the population in that river. So it's the thing that the characteristic is whilst that river maintains its legacy pollution, the fish within it are quite happy. But if there's any change, it's like they've got a very small hand of cards and they've got no way to respond. And that's the danger with populations that have become adapted to these unique kind of pollutant pressures. It's that they lose a lot of their diversity in order to be able to respond, and it leaves them hugely ill-equipped for any future change in their environment. And I think that's the thing that we notice most drastically is this reduction in diversity.
Thank you so much.
I hope that was useful.
Yeah, the answers it perfectly. I'm sorry, but due to time, we have time for one more short question. So, yeah.
Oh, I figure the microphone's here. So this is probably a stupid question because I'm not a biologist. Is there any scope for using environmental DNA? And could you connect in these demographic type studies? Could you link the eDNA you observe in a river to a population, or is there just not enough information in it?
Shall I pitch in?
Yeah.
Yeah. eDNA is improving. I'm from a family of people that have been quite skeptical about eDNA. I think there's a time and a place. We're seeing, for instance, eDNA used in assessment of bycatch in the holds of fish to check whether there were salmon present and things like that. I think we've moved beyond a presence absence, a yes no, but a lot of the markers that are used for eDNA will typically not entirely, but a lot of them still center around mitochondrial DNA because of the protection that mitochondrial DNA offers under environmental degradation. If you were to use that, you would lose some of the possibilities for that fine scale resolution that you get with, say, SNP markers, which are drawn from across the genome, 27, 29 chromosomes in salmon and 40 chromosomes in trout. So I think I would be interested to try it, but I wouldn't want to put too much accuracy on any major management decisions based on eDNA results at the current time.
Great, thanks.
Okay, and I think that now already we don't have time for any more questions. Thank you so much for the presentation. Thanks for the audience, and apologies for the connectivity problems at the beginning. So thank you so much.
Thank you.
Thank you.